Hi, everyone. Welcome to Spatial Omics Seminar Series 7. And today we are very excited to have Dr. Fiona Ginty from GE Research. And just briefly, Dr. Ginty received her undergrad degree from National University of Ireland, Galway, in microbiology. After that, she received her PhD in nutrition sciences from University of College Cork. And finishing her PhD, she joined Nestle as a postdoctoral scientist in the Minerals Research Group. And following Nestle, she joined Medical Research Council in the UK to study bone in the nutrition research. And after that, she joined GE Research, first as a project leader, and now she is recently a biosciences technology manager and principal investigator. Briefly, if you remember the spatial proteomics papers back in the days, their demonstration was the very early demonstrations in the field, the PNAS paper and many others. So today we're very excited to learn about the recent data and then the most recent platforms that they're developing. And without further ado, I'd like to then turn over to Dr. Ginty. It's all yours.
Great, thanks, Ahmet. Thanks, everybody. Can I ask you if you can see my full screen? Just if I go to my first slide, can you see everything? Because
yes,
I can't. You can see it. Okay, great. All right. So just very, very quickly, just wanted to first introduce where I am. Not everybody is aware of the GE Research Center. I see some folks on the phone who are actually worked here at one point, so that's nice to see you. This is a multidisciplinary research center. We work for General Electric Company, but about half of our time is also spent on external projects. We partner with academic groups, key opinion leaders, clinical centers of excellence, national labs, and small businesses and companies across the US and globally. And a lot of different technology domains you can see there on the left. I'm in the biology and applied physics lab, but a lot of our projects entail working with people across the center, which actually is a very nice thing from the perspective of new ideas and new areas in which we can work. So a lot of cross-disciplinary work, a long history of partnering with the government and NIH, DoD, DARPA, etc. And with that, we also have the opportunity within this environment to sort of think from an applied perspective. So we do a lot of basic research to figure out the tools and methods, but also have a purview to look down the road and see how can we potentially translate things to products. Now, not everything we do turns into a product, of course. We do a lot of research that continues via NIH, etc. So the little picture there of our front door, and then just the geographically located, since we're talking about mapping today, we're right about 3 hours north of New York City, about the same from Boston and South Montreal. Very, very briefly, our group, my group, I'm in the biosciences organization or lab. We work closely with other imaging radiology groups, MRI, X-ray, PET, CT, ultrasound, physiology monitoring, and we also have a project in bioelectronic medicine. So again, most of our work in the biology space, we work in our group, we're concentrated with biology, molecular biology, cell biology, pathology, of course. And then we often try to intersect where we can with the other modalities in MRI and X-ray in particular. So I'll get right into the topic for today. And this is a topic that we've been working on for some time, although some of it we sort of stumbled into, I would say as well, because at the time when we developed our technology for spatial cell analysis, it was back in 2007, actually, we started working on it. And at the time, the clinical needs, although they haven't changed, were more around the automation of the workflow, the standardization of biomarker quantitation, and the ability to analyze multiple markers in the same sample, thereby saving sample. And as we developed the tools and worked with groups over the years, and of course the whole field has changed with DNA sequencing, of course, as well. But the major components, of course, these are not the only ones, but just to kind of draw on the things that we probably have the more experience or exposure to, are covered here in this slide. So, you've got the different cell types, you've got vasculature, hypoxia, different cell states, all of these in combination. They may, one or all may be present at any one time. And of course, that makes cancer research particularly challenging, but also very interesting and exciting. And I think the hallmarks of cancer, which were recently updated to include some additional components, including microbial influence. But it's a nice way to think about tumor heterogeneity. Not all of these components will necessarily be in place at any one particular time, but it's a way in which we've tried to structure our thinking. And as we've worked, with other collaborators, they've helped us think through these problems. So I think the one thing I'd like to sort of get through today is just that sort of the systematic thinking of how we've tried to think through tumor heterogeneity. We haven't solved it. It's still, in terms of, the meaning, the value, and the clinical implications, all still need to be figured out. But hopefully I can share some of our experiences and how we've approached this. So first is, a lot of our research to date, and in general, I think in the field, has been focused on tissue microarrays, the small tumor cores, typically 1 to 2 millimeters. They can be a little larger, but and there's a trade-off, because of course it's easier to do a lot of patients and do a high throughput analysis on a lot of cores and patient cores. The trade-off is, of course, that you miss out the bigger picture, and so that is what it is. And of course, you would like to be able to transition to whole slide sections to translate your findings where you can. But this particular slide shows, really the point here is to show the extent of variation that you can see across a tumor and the surrounding microenvironment. This is stage 2 colon cancer, part of a ORA1 project that we're doing. And the components that are of interest and the things we've studied are the tumor microenvironment, the immune, the tumor stroma, and the tumor itself, the tumor center. I pull on this because this is not a tumor. This is from a company in the UK that does what's called digital geography. And I always like to think about geospatial as sort of a reference point as we think about things, because there's so much done in that space with regards to spatial analysis and context. And one of the big things, of course, is when you build a map, you of course need the right measurement tools and the local knowledge. And depending on what you're trying to build a geospatial map of, it could be a city, it could be infrastructure, it could be forestation, it could be floodplains. But I think many of the things that we think about in that context, although oversimplified when it comes to tumor, but it's a way to sort of structure one's thinking. At the research center, we've got access to people who work in many different domains, and I show this example here. I am not a power distribution network expert by any means, but when we first started doing the spatial analysis and thinking about it over 10 years ago, actually the folks who were working in the geological space, in the sort of the mapping space, were the first to help us in terms of thinking of tools and platforms. So this is just an example of where you're going from, and it's kind of similar in the tumor biology as we try to think about it. You've got to go from sort of very granular detail, in this case, substations and all the components. Then you've got the network across the US, and then really at the end you have to bring it down to local level and see what the power needs are in your local vicinity. And then, so taking kind of the similar paradigm in terms of tumor biology, there's similar level of complexity, of course, way more variability potentially. And you've got the genomics, multi-omics, really, the tumor cell biology, the tumor itself, metastases, and then depending on what your endpoint is, it could be progression, therapy response. So working at each of these levels requires, a slightly different approach, different sample size. I won't talk too much about this end-to-end process today, but will mainly give you flavor of the pieces around the tumor cell analysis that we've been doing. So with the spatial biology, of course, everybody who's on the phone today has probably been touched in some way by spatial analysis, either spatial transcriptome, spatial proteins, and no doubt it's revealing many new insights into tumor biology, microenvironment, cell types, new disease mechanisms, so much potential in terms of what we can do, and it can be definitely overwhelming in terms of figuring out what the right questions are. Many advances, especially in the last 10 years. This is a really nice review from the end of last year. This is from HubMap, which Human Biomolecular Mapping program that's part of the NIH, funded by NIH Commons, and we're responsible for skin analysis together with the University of Pittsburgh. And one of the goals of HubMap is to bring together all of the different methods into one space to do analysis of all the major organs. And with that, we've got a big emphasis on streamlining our workflows and standardizing them and making sure that they are done in such a way that we can compare across organs. So this is a really nice overview that gives a summary of all of the different methods that have been developed in this space over the last 10 years in the protein space. So you call it fluorophore, which is where cell dive, which we developed. You've got the DNA labeling methods, Codex, Immunosaber, and the metal labeled methods, which is MIBI. And then, of course, the spatial transcriptomic side has been exploding as well, and many exciting methods also in the running there as well. So another nice recent paper published at the beginning of this year, looking at different methods, comparison of different analysis methods for each of these different platforms. And then with that, there's so many different atlas mapping efforts that are happening right now. HubMap, which we're part of, you've got the Human Cell Atlas, the Human Tumor Atlas, you've got kidney maps, and then SENNet, senescent cells, lung map. So it's really bringing all of these tools together in such a way that hasn't been done before. And I think over the next 5 years, we'll see an explosion in terms of data, single cell data, from all of these different types of modalities, in addition to combination with lipidomics, proteomics, metabolomics, etc. So exciting time to work in single cell analysis. And then we see advances into other diseases. This was a really nice paper that was published at the end of or during 2021, looking at COVID infected samples or patients, and the interestingly, a lot of different analysis done in this study. One of them was looking at the spatial relationships between COVID infected cells and different cell types. So definitely worth a read. There's been several more papers since then, but I think this was one of the interesting first ones that came out. So in terms of mapping tumor cell biology, sort of with that backdrop, would like to break it down into sort of 4 key components. Definitely not restricted to these. This is a reflection of what we've done, and I think there's many interesting papers that are on top of what I'll talk about today, but this will give you a flavor of the kind of things that we've been doing. So I'll just start first with the cell dive workflow, and some of you are already familiar with this, so I won't spend long in it, but it's an iterative staining method. We reuse the same sample n number of times, stain over 60 biomarkers in the same sample. We've developed the technology here at the Research Center. We continue to work on it from a research perspective, but it's now commercialized by Leica Microsystems. So that was nice for us, nice for the team to see it transition into the real world, as they say. Few sort of components just to consider when we do cell dive analysis. There's distinct rounds of staining, so distinct from maybe other methods like Codex, for example, which does all its staining in one go. We have iterative rounds, so it gives you some flexibility in terms of the types of markers you can swap in or swap out. And all the image processing is done online as the imaging is done. So a few key steps: the registration, autofluorescence removal. And then finally, as you sort of get past all the hard work of doing the actual sample analysis, now you've got potentially millions of data points to analyze. And so just some workflow examples here. You've got your multiplexed images on the left. This comes from a colorectal cancer study we did, and it's published in PNAS. Segmentation, a key part of the workflow, segmenting into epithelial cells, stromal cells, immune cells. Certainly possibilities to extend that even further, depending on which markers you've included in your panel. And then combining the biomarker with the segmentation, you now have data set that has biomarker intensity at cell level, plus the spatial location within the sample, and then the cell ID as well. So that data set essentially gives you the possibility to start to look at questions that are spatially focused, and/or you can look at average expression across the sample. So many ways to do the analysis. So we very simply, some of the types of things that we would do, especially when we would start an experiment, and some of this is very routinized in the sense, when we first started doing single cell analysis, it took an extremely long time to process the data with a lot of noise. So we learned along the way in terms of controls and filtering criteria. We'll touch on some of that towards the end of the talk. But one of the really useful first steps is looking at correlations between biomarkers, and that gives you a sense of how well things that should be co-expressed are correlated with each other, and are they negatively correlated or not correlated at all. One tool that we use a lot is k-means clustering. It's been a really nice method for identifying groups of cells with similar biomarker profiles. And we've used that extensively over the years in different projects. And I'll talk about one of those today. That's the work we've done actually with Sunil, Dr. Babbi, and Yassine Polo are on the phone as well. And yeah, once you start to do the clustering, then you can apply the clusters or the membership of those clusters back to your sort of population cohort and project it back onto the images, and look at the distribution of different cell clusters across your population. Another area that we've worked on, and this is probably very much expanded since we started to work on it, but is looking at cells, spatial heterogeneity, cell neighborhoods, and distances between cells. And this becomes extremely interesting and useful when you start to look at tumor edge, immune cell interactions, etc. So with that, I'll start to talk about some of the areas that we worked on in terms of tumor cell composition, the microenvironment, immune cell response, and functional state. So one of the very first projects that we started, like all of our work on, was in colorectal cancer. And this was sort of our hallmark study, if you like, when we first started developing the technology, and we conducted analysis of over 600 patients with 60 biomarkers. And this is where we started. Chris Savinsky actually was in our team at the time. He really started to think about heterogeneity from a colorectal cancer perspective. And now seeing these layers of images, started to observe some of the variability that is evident once you start to, of course, look at serial sections. It's evident, but now you start to overlay it on each other, and then you really start to see the diversity. So one of the first publications on this was part of the PNAS paper, and here in that cohort that I just mentioned, we looked at, found very distinct groups with regards to mTOR and MAP kinase pathway. Not correlated with outcome, but got us sort of thinking at the time, perhaps this was related to drug therapy implications and so forth. But it I think one of the challenges in these types of studies is, not having either therapy or having a sort of a clinically actionable endpoint. And if you are simply just looking at prognosis, particularly in colorectal cancer, you also want to know, is a patient going to respond to therapy? So we took this analysis one step further. Our collaborators at UPIT led this work on the same cohort, and they found, took this to the next level in terms of spatial analysis, and this was published in 2020. And they took it, I think it was very interesting what they did at the time. It was not just focused on single biomarkers, although we had the biomarker information for 55 biomarkers, but they looked at it in terms of correlations between biomarkers and within the epithelial domain, the stromal domain, and then the epithelial stromal, the boundary, that 100 micron boundary between the two. And once they started to look at it from a correlative sense, like which biomarkers were most strongly correlated with each other, and using that in a sense as a feature, that started to reveal some interesting biology. And they took that one step further by looking at spatial domain network analysis, looking at the different pathways that might be correlated with these features, these correlative features that they found to be very strongly correlated with outcome. And I'll jump around a bit here as well. I'm not necessarily following a theme for one particular cancer, because we were, been fortunate to work on many different cancer types, but I'll give you some flavors along the way of our findings. This was another study that we did with Memorial Sloan Kettering on tumor cell composition and FDG uptake. And here we were really trying to understand what was the MSK had previously done work in this cohort looking at RNA-seq analysis, and found particular profiles related to the glycolysis pathway and bulk expression, and how that was related, and found that that was related to FDG uptake. And that's not surprising. FDG is a glucose analog, and of course it's going to be more likely to be uptake by cells that are have high expression of the GLUT1 protein. But it hadn't been done at a protein level before, and not in a sequential, in a 5 micron section. So here we found very similar results to the bulk RNA-seq analysis, in that the FDG uptake was correlated with, particularly at a macro level, hormone receptor negative tumors, so ER-negative tumors. And probably the next slide just shows you a little bit more color in terms of the diversity. So higher tumor cell metabolism, high proliferation and PTEN suppression in hormone-negative tumors was independently correlated to high FG uptake in breast cancer. And what we observed in this study was, it was just both interesting as well as biologically, mechanistically sort of proved out some of the value of doing these types of analysis at a single cell 5 micron section, even though from a resolution perspective, the PET resolution greatly exceeds what you would find within a single section of tumor. But we were still able to find this correlation. So indicating that, yes, you will find, there are pockets of disease within a TMA, or that are reflected within a TMA or in a whole tissue section, but it's scaling to the whole tumor. I think that's an unanswered question. Can you elucidate from a single section, what's happening in an entire tumor? But it's still interesting that you can see these correlations between, the phenotype of the cells and glucose uptake at a radiology level. So moving on to the microenvironment and immune cell response. So I'll just cover both of these together because in the interest of time. And first off, I think, one of the advantages of multiplexing is being able, of course, to do as many markers, within a particular pathway or cell type. And here we see, this is work that we did, again, this was in the colorectal cancer study that we did early on, and just the diversity of immune cell types, the tumor cells, B cells, macrophages, cytotoxics. It's incredible to see from a patient-to-patient perspective how much variation there is. And then there's, of course, the blood vessel aspects, and I think this is a little bit more complex. There are some really nice studies that show vasculature associations with outcome. It's hard to accurately segment blood vessels in that they tend to be, you're seeing a two-dimensional version of a fraction of a three-dimensional structure, and so you're seeing pieces of the blood vessel. There's also the extra level of complexity. This is work that we did with Eli Lilly on the maturity of the blood vessels. And so one project that we did with them, with Eli Lilly over a few years, was to look at different markers that could be used for blood vessel segmentation. And that would sort of do twofold. One would inform about the maturity of the blood vessel, and two would give a better segmentation, so more complete segmentation of the blood vessel. And we've taken this further. This is not a tumor, but this is skin. This is the work we're doing in HubMap. And we've been looking at the distances from endothelial cells to sort of nearest neighbor immune cells. And I think these tools that we have been working on, this is with Indiana University, these types of tools can be extended into tumor biology as well. And I think would be, certainly be very informative with regards to different cell types and different different tumor cell types. But there are considerations when profiling immune and blood vessels. We talked a little bit in terms of the continuity of the structure, the expression, the benefits of including other markers of associated with the endothelial cells. And similarly, so with the immune cells, you have ideally you want to have consider multiple markers to have higher degree of accuracy for your phenotyping. So, for example, the combination of CD3, CD4, FOXP3 for the regulatory T cells. Macrophages would benefit from many 2, 3, 4 markers as well, and of course then you want to layer on functionality as well. One challenge that can certainly be the case with the immune cells is there's a wide range of signal and background, so a single threshold may not work. And there are some really nice new deep learning methods and machine learning methods that have been developed over the last few years. So some of this can be worked around, but there are also certainly some challenges to consider in terms of signal and background. And having additional biomarkers to confirm the identity of a particular cell type can be extremely beneficial. You have cells that don't have well-defined boundaries, and that can pose additional challenges as well. And then you can have overlapping cells that are very closely compacted with each other. So separating those cells is of importance so that you don't do a double positive or false positive expression. So this is one of the approaches that we've taken. We've developed a machine learning based method for immune cell classification, but we've evolved it over the years. So some of the methods that I'm going to show you are the methods that I'll talk to you about right now, or focus on that machine learning method where we consider both the cell boundaries and as sort of our starting point for classification. So that gives you a higher degree of false of accuracy. So you set a boundary that your signal for that particular protein is then overlaid with. And you can then train the algorithm based on your annotations, and then combine every cell. Then the other benefit of using the cell as your sort of boundary point is that you have the spatial location and the ID for already generated for that particular cell. So that allows you then to incorporate the immune cell data with the rest of your data set, the tumor cell data set. So this is work that we did with Dr. Vadzi and Dr. Poehler at Indiana University, now at Emory. And this was a really nice project we did together over, it was a ORA-1 over 5 years. And we looked at different facets of DCIS biology, both from a progression perspective as well as heterogeneity. And this particular study I'm showing you here, this was done in whole tissue sections. And here we were trying to understand twofold. One, just a general landscape of what the immune response was to in different patients with DCIS. And then two, how many images would it take to capture the heterogeneity within the samples? And that's an important perspective. Especially if you're working with TMA cores, are you capturing the heterogeneity within the sample? So 2 patients here, just for contrast sake, the upper one has the multiplex staining. So we've got what we call the virtual H&E, and the first panel, that's the it's the autofluorescence image that's converted to pseudo-colored to the purple, pink colors of the standard H&E. That gives you your sort of starting point from a reference perspective. And then multiplex imaging, the cell clustering, so the k-means clustering output. And you can see in the upper one, you've got sort of more concentrated one particular cell phenotype, whereas the one underneath it has much more mixture of different types of cells, and then immune response quite different between the 2 patients. And so one of our outputs from this paper was to demonstrate that you would need, in order to capture the heterogeneity in this particular study, you would need to have 3 large TMA cores of approximately 2 millimeter size. So segueing into the functional state, and here I'll talk about the work that we've been doing with on colorectal cancer. And this is a project that we've been doing over the last 4 years with Royal College of Surgeons in Dublin, Queen's University in Belfast, and Memorial Sloan Kettering here in the US, and ourselves. And our goals were to develop a workflow by which the team in RCSI had developed a systems biology method workflow for calculating apoptosis sensitivity using the input of 4 or 5 proteins to estimate the MOMP and other downstream activation of the apoptosis pathway. So we had 2 goals in this particular study, which we published last year. We wanted to generate an atlas of cell states in relation to apoptosis sensitivity. And we also looked at spatial analysis of these of apoptosis sensitivity of the cells in the particular sample. So how spatially were they organized? Were they more distributed towards a particular cell type, etc. So one nice thing about this study is that we incorporated cell line controls, and this is again another point from a quality control perspective. The cell lines were important in this particular study to provide an absolute or a quantitative reference for each of the apoptosis proteins that we were that the team in Dublin were incorporating into their model. So they wanted to have a reference that the cell lines generated that reference. They had previously quantified the amounts of expression of these proteins of interest within these cell lines, and so that gave them a reference on which to calculate the amount of protein expression within the tumor samples. So we had 1,300 patients across 37 TMAs from 5 institutions, and so a couple of other considerations that we had to employ here were batch control, normalization. Again, sources of noise are not friends when you're looking for heterogeneity signal, and so we took great care with in terms of the calibration and the inclusion of controls, and then staining in single batches, and so forth. So in terms of our workflow, pretty simple in terms of what we started out with. We employed our standard workflow. We did an epithelial, stromal, immune cell classification. So that gave us our sort of baseline map which to start with. The team in our CSI then calculated the apoptosis sensitivity for all cells. And then because we had the spatial location and the ID for every cell, we were able to assign that to it, whether it was immune cell type or tumor cell or stromal cell. And so the image that you see here is just an example from that paper. Shows 3 different patients, and then the staining of the tumor of the immune cells, and then the projection or underneath that the sort of the digital map of those cells within those images. And so then on top of that, we then projected the apoptosis sensitivity on top of that. So this shows in the next slide where we looked at the diversity of apoptosis sensitivity across these tumors. And the image on the left shows the patients that were sorted by stress dose, so from going from low to high. And so on the left with the red and the gray image, that just shows you your cell classes. So you've got cancer and immune cells and stroma in red, blue, and gray. And then projected on top of that, you have what they call the Dr. Momp score. And so that would be sort of the higher the score, the higher the sensitivity of the patient to apoptosis. So and then so on the left, you have patients who have who are on the lower end of the scale, and then on the patients on the right, you have the patients at the higher end of the scale. So this gave us a really nice demonstration of the ability to combine both the computational model for apoptosis to calculate sensitivity to apoptosis together with cell type. And what we found was, which was agreed with earlier research, so it was sort of a validation, was that the immune cells had the highest sensitivity to apoptosis, and then similar between the cancer and stroma, but some variation, of course, within each of those as well. So what we've done since is, now in this study, we did not find a correlation with outcome. So we looked at heterogeneity using several different methods that are, been used in other contexts. So we did not find a correlation with outcome, but our sample size was relatively small. So we've since taken this further into the 1,300. So this particular study was done in just 140 patients, I believe. So now we're looking at the 1,300 patients, stage 2, stage 3. We have more cell markers and more cell type markers. So we're doing a similar type of analysis, but in a larger sample and with outcomes. And so we'll, over the next 6 months, I hope, we're right in the middle of the analysis right now and starting to see some interesting results in terms of some of it is validating what we found here, but we're also have other information around the metabolism of the tumors. We have blood vessels, so we're able to hopefully create a map of that incorporates the other environmental factors within the tumor as well. So then that sort of gives you a snapshot of the different studies that we've done in heterogeneity. I've gone pretty fast through them, and happy to take any questions, but we've approached heterogeneity, as I said, from both a pathway perspective, spatial, considering the tumor microenvironment, the tumor cells themselves, the infiltration of the immune cells, and then the consideration of the vasculature, and there's other components as well that I didn't have time to go through. But you can see there's so many possibilities in terms of if you wanted to take this to the next level. Our focus was more clinically focused in terms of analyzing progression of disease, risk of recurrence, but there's many deeper studies you could do within each of these studies to look further at the relationships, neighborhood analysis, and I know Ahmet has done some really nice work and has developed some tools in that space as well. So some practical considerations when you're thinking about heterogeneity and planning this kind of analysis. We started out at the very beginning of the picture of the colorectal cancer tissue with wide degree of variation across the sample, depending on where you sample. Are you looking in the right place? Are you asking the right questions? That's one benefit of, as I said, of tumor microarrays is that you can analyze a lot of patients, but your results potentially could be biased in terms of from where the sample is taken from. So ideally, if you can do whole sections, I think you certainly got a lot more real estate to analyze, but you'll still probably have to narrow it down to regions of interest that are of relevance to the question you're asking. And so with that, the same point, ideally, if you are doing tissue microarrays, you've got multiple cores per patient, replicate sections, balanced TMA design. So are you mixing your stage ones? If you've got one slide for stage 1, one slide for stage 2, one slide for stage 3, that can be challenging from a normalization perspective and an integration of data. So ideally, if you can mix the different stages on the same slide, that would be ideal. We didn't talk about antibody specificity today. That's certainly goes without saying that the sort of the rigorous workflow to ensure that your antibodies are staining what they're supposed to be staining. We over the course of the last 10 years, we've validated over 500 or 600 antibodies. Spent a lot of time and money on antibodies that didn't work. At least we did it upfront. But even when you have an antibody that can sort of pass all the criteria, if your sample is of poor quality or old, you can have other sources of noise, depending fixation time, you can still have some issues with background. So it's something to keep an eye on, especially if you're doing any type of analysis that involves thresholding. So you just want to watch out for that. Segmentation, there's so many different methods now for segmentation. So it's, I think the space has really grown. We've evolved, we've developed our own cell segmentation workflows, but we also like using QPath. It's a really nice workflow. And then I know through HubMap, there's also tools that are going to be released as part of that as well. So I think no shortage of well-validated segmentation analysis tools that are coming out. One important thing to consider is just screening for imaging artifacts. I'll show you an example of what I mean in a minute. If you can include cell line controls, that's great. They do take up space on your slide, so you have to consider what your experimental question design is. And there's a sort of finite number of cell lines that might cover all of the markers that you might include in your study, but minimally including a positive control sample, a reference sample that you're confident stains all the markers that you're interested in is important. I just added the last point at the end about radiology and pathology, because there are a lot of opportunities to compare heterogeneity at a tissue level with radiology. But one thing to consider is just the difference in scales. And I'll just show you what I mean by that in a minute. So just in terms of some examples, if you have low abundance cells or weak biomarker expression, that can be a challenge, both say from a clustering perspective and just in general, identification in general. At least, there are many examples of rare cells or and so forth. So you may not even be, it may not even be a concern to you, but we have examples where we've, in general, we see low section to section variability for most of the markers that we look in, but where you do see higher variability, it tends to be where there's only a fraction of cells positive, or where there's overall low signal. So again, just something to bear in mind is when you're doing any kind of heterogeneity or spatial analysis, that itself can be a source of variability in your results. Normalization, definitely an important QC step. In this particular study that we did with colorectal cancer, there was 37 slides, 5 institutions, and our goal was to integrate this data to the extent that was possible, and especially use it for validation and so forth. So in general, it's not this is not our finding. We know that normalization across multiple slides is needed to reduce batch effects. What helps that is if the patients are randomized on each of the slides. You don't want to eliminate any true biological signal when you're doing this kind of normalization. So recognizing this as a source of error, we took it upon ourselves 2 years ago. So John Graff on our team evaluated all of the different normalization methods that have been used in different contexts, not just in fluorescence imaging, also in RNA analysis. And he developed a nice method which you can download on GitHub it's open source. So it's a fluorescence image, so we call it FLINO or FLINO, fluorescence image normalization based on upper quantile. And so we apply a grid, so we don't use the cell level data to generate the normalization process. We apply a grid over the entire sample, and that then is used to do the normalization, so that we're not relying on, we're not eliminating any, we're not including sources of potential noise, or we're including all the data that's in the image. But what can be challenging is if you segment at cell level, you can have biases in terms of one cell type or another. So this is sort of unbiased based approach, and so your link to the paper there is if you're interested in normalization. I know it's not the most exciting topic, but it's an important one if you're interested in heterogeneity for sure, and any type of analysis. And then artifact detection. This is also an important source of error. You can certainly mess up all of your heterogeneity analysis if you have imaging artifacts. And this is just an example. These are examples of, on the left, Sang-Hee Cho, who's our statistician in the group. She generates these intensity plots. So this helps us because you generate massive amounts of data when you're doing multiplexing, and so you can't physically go through each one of them independently. So these intensity plots are a way in which you can sort of screen out anomalies within the entire data set, and then go and visually check them as well. So in this case, it was an imaging artifact. So we can either screen out that region, so you don't lose out you can screen out all those pixels, so you don't lose the entire sample. And then just my last point is just in terms of comparing radiology data and pathology. There's certainly, we know this, they're on different scales of resolution. CT, you've got about 500,000 cells per voxel. MRI depends on which magnet is used, but T1, my MR colleagues have educated me, it's 1 million cells per voxel. And then when you're looking about PET, it's much lower resolution. So you're talking about 27 million cells per voxel. So it can be challenging then to collocate biopsy sample or information you see in a tumor sample with radiology features. So either custom design studies may be needed, or you can just take the approach that this is a correlative analysis, and you're looking at it from the tumor biology from 2 different perspectives. So just coming up to the last slide. So just things I wanted to communicate today, and hopefully it was helpful. So for sure, try and define your questions up front, because that will inform your panel of markers. And the last thing is how it happens every time you get to the final question, you say, I wish I had that particular marker. So that will happen, of course, and then you have to maybe go back and redo the study or think about it a different way for the next time. Many analysis approaches. I just talked about some of them that we've done today. I've talked about segmentation, I've talked about k-means clustering, but there are also many other approaches in terms of distances and distance analysis, neighborhood analysis, social neighborhood analysis, and so forth. So, these are all really interesting approaches for looking at biological questions, especially in relation to immune response, tumor interactions, tumor cell types. And then we touched on apoptosis as one measure of cell state, and you could imagine similar maps being applied for other cell states as well, proliferation and so forth. And then finally, good quality control, of course, is essential, biological and technical. And then just to acknowledge, so some of the projects I touched on today, the work with the Sunil and Yasim on an NCI provocative question that was in DCIS, and happily, we still work together and enjoy that collaboration very much. Colorectal cancer, that's work with the College of Surgeons and Belfast, and then an MSK. And then finally, the HubMap project with the University of Pittsburgh, where we're looking at skin, healthy skin, and looking at different tools to do 3D reconstruction of skin. I didn't have time to talk about that today, but another interesting project in terms of spatial relationships in 3D. And I think that was it. I had some backups, but I'll take questions if you want to, if there's time for that, or happy to, if anybody wants to send me an email. My email is on the first slide, so I can share that afterwards, or Ahmet can send it out. So thank you, everybody.
Yeah, thank you so much, Fiona. All right, so whoever wanna ask a question, you can actually just unmute yourself and ask. If you want to put in the chat box, that's totally fine as well. Okay, maybe I can start with my first question regarding the vasculature identification segmentation. That's very hard in the I think often just a one section of the tissue we just cannot see where the vascular network is. We often just say, okay, a few locations you have like one blood vessel, but that particular sample looks like a lot of microvasculature, the capillary there. That's kind of quite interesting. Also, I don't know how you did your segmentation, but I noticed you have the collagen 4 staining. So it turns out collagen 4 is the kind of extracellular matrix protein produced by endothelial cells. That gives you a very nice sort of vascular network structure. I don't know, how did you do that? Did you actually use the collagen 4 to do vascular segmentation? If that's true, that's probably the better way to go, indeed.
Yeah, I mean, you can get it, certainly. I think the combination, if you CD31, CD34, collagen, and smooth muscle actin, those are the 4 that gave us the kind of, the combination gave us the much better resolution on the vascular structure. Of course, collagen is staining other things as well. So the algorithmic approach basically combines all 4. So it's sort of ignoring anything that's not positive for those other 3 markers, and that gives you a much nicer vasculature, much more resolved structure of the blood vessels. Yeah.
Yeah, thanks. Yeah, there are 2 questions in the chat box. If you can rate yourself, I think, Fiona, that's, yeah, you can just go ahead and answer those questions. Otherwise, I can read for you as well.
So yes, I see one from Christian. Published evidence, considering the increasing published evidence of tumor-associated bacteria, do you think it was possible to localize bacteria in the images? So yes, I absolutely. I think that's a very interesting new area. I mean, it's not new, but it's just in terms of the ability to multiplex, and there are some very nice fish-based methods that have been published in the last few years for detection of bacteria. We have tried some different approaches with H. pylori antibodies, for example, and I think it needs some optimization because you have to work at a higher magnification, and there, but I think it's certainly possible. So, and I think that would give you some interesting insights. Yeah. So do you, as a group, do you research mostly supporting internal R&D work or providing service to external groups, or both? We do a blend of everything, Byron. So we, our group supports GE businesses that are in product development mode. So we, the next generation of CT imagers, for example, whatever. But we also, about 30% of our efforts, the research is externally focused, and including NIH, DARPA, DOE, ARPA, so across the whole sort of range of industries, including our technologies. Rather, we work very closely with external groups, and so we. And service, it's a smaller part of what we do, but I'm happy to, if you want to reach out. It's the cell dive itself is offered as a service. We do our own research on it as part of the NIH, but it's Neogenomics in California does. It's under the name of Multi-Omics, and they provide service for cell dive analysis, just to distinguish that as well. Do we use fluor antibodies? We use, yes, I didn't go into the detail, but we directly label the antibodies with cyanine dyes, Alexa dyes. So that's cell dive. We can use secondaries as well. So in the first round of staining, you can use primary, secondary antibodies, and then you're just species limited after that. So that's why we directly label all of the remaining antibodies with Cy2, Cy3, Cy5, depending on what your imaging system is. I think the upcoming or the current Leica microsystem cell dive has Cy7 channel on it as well. So you could do Cy2, Cy3, Cy5, Cy7, and you could do that in sort of an iterative way. Lots of questions. Thank you. Have we looked at integrating CT and MRI and cell data? So with the skin study that I mentioned that we're doing a HubMap, in fact, we are doing CT imaging, micro CT imaging of the skin cell blocks or the skin tissue blocks. So we do micro CT imaging of the block. So that gives us a 3D image, and then we do sequential sectioning of the block. So we section it up to 100 times, and then we take about 26 sections, and we use the CT to guide the reconstruction as a reference for reconstruction, for 3D reconstruction of the skin samples. So yeah, that's another talk for another day, maybe. But so we have combined micro CT and cell dive data from that perspective, and then we've also correlated features within MR images with cell dive data as well. That's a study we did a couple of years ago on glioblastoma, and so you can find that online as well. One more. Have we done single cell RNA-seq in combination with? So we're planning to. So yeah, good question. Definitely want to get that incorporated into our workflow so that we get generally richer data set in terms of cell type, and then combine that with cell dive spatial classification. That would be definitely something we're hoping to do over the next year. And then in terms of normalization, is it possible to normalize using a total protein dye? Yeah, I think that could be useful. The only challenge may be that you've got, it's sort of an artificial construct, so you don't have the kind of background that you would have in a tissue sample. But if you yeah, I think it's worth thinking about, but just some considerations in terms of the background signal and the autofluorescence that comes from a sample that you might not have in sort of an isolated reference. I think I got everybody. Thanks. They're great questions. Thank you. Did I get everything wrong?
Yeah, I think so. I think all questions in the chat box I can see.
So I think great question, Fiona. Thank you. So I'm also curious about the general trends right now, right? There are many proteomics technologies coming up. What is your vision for the future? Are we moving into, let's say, spatial proteomics soon, or how do we bridge the gap?
So vision for the future in terms of clinical
Spatial
applications, Ahmet?
Clinical and technology, of course. Mm-hmm.
Yeah. Yeah, I think the shift is going to happen, or needs to happen, towards whole tissue imaging. I think the small tissue samples are really good for discovery, but they don't give us the complete landscape. You know, I like the digital geography example. I mean, you still have to hone in on your region or question of interest, but potentially miss out on a lot if you don't have the whole tissue sample. So I think that's an important direction. I think in terms of image processing and analysis time, you know, that can be slow. I think, you know, we have to rely on high performance computer for some of our, very, you know, much more in-depth analysis. And so that's not necessarily something that everybody has access to. And, but in terms of like turnaround, like, you know, before we started doing high performance computing on our analysis, it would take maybe 2 weeks to sort of turn around from a question, you know, iterate. So that's, you know, you really want to go faster than that, I think, because you sort of, you know what you want, but you need to see it again and then again and again. So I think you have to, yeah, the speed of analysis is going to become, is like already, it has to be faster. And I think then too, the visualization of the data, just it's complicated. You know, we've been thinking about this for a long time, but when you're new to the field and just trying to get all the permutations of the things that are up or down or correlated. So yeah, having better tools that combine, sort of visualize your results and then combine it with other omic technologies, like somebody mentioned the RNA-seq, single-cell RNA-seq. You've got the spatial transcriptomic methods now. So, I mean, you don't need to have everything combined, but I think not everybody will have the possibility to do that level of integration. But, you know, as we want to gain more and more confidence with these multiplexing approaches and the value of spatial proteomics, you need to have, I think, better tools for visualization of data. So those are 3 thoughts. I probably could think of more on it, but hopefully that gets you.
Wonderful. Yes, I agree. Yeah, I think there are a lot of opportunities. Wonderful.
Yeah.
Are there any other questions? Then, if not, let's then thanks to the speaker again. Thank you, Fiona, for this fantastic talk. I'll also use these signs filters. So here, I found it. Thank you so much.
Thank you.
Appreciate it.
Appreciate it.
And
thank you, Fiona. Very impressive.
And before
and thank you to the fantastic teams that we have at the research center as well. Definitely not just me. There's a much bigger team that I work with.
Wonderful. And thank you again. Before we wrap up, I'd like to also announce that we will have next week Dr. Dana Pierre from MSK talking about computational approaches to mapping structure in tissues. And with this, thank you, everyone. Thank you, Fiona, and hope to see you all next week. And have a nice weekend.
Thank you very much, everybody. Bye-bye. Bye-bye.
Have a good weekend.
You too. Happy Friday.